Okay, good afternoon, everybody. And this afternoon, we're going to carry on with our discussion about DNA, RNA, all of the other nucleic acids and their function inside the cell. And I just remind, this is where we ended off last time. And I just remind you of this really important experiment, which demonstrated that, in fact, DNA was the hereditary material. The DNA was able to confer genetic information, and this was demonstrated by Griffith in this experiment on Streptococcus. I'll just briefly repeat it for you because it's so important. He took living smooth cells of Streptococcus, which are pathogenic because they have a capsule, and demonstrated that they could cause disease in mice. He then took another strain of Streptococcus, which was rough, which has no capsule, and it does not cause disease in mice. He then took his culture of the pathogenic bacteria, he heated it until he killed all of the cells. But when he mixed that together with the rough, he found that it was able to kill mice. And when he extracted bacteria from the mice, they were smooth. And what had happened was the rough bacteria had acquired DNA. When he heated the smooth, killed them, DNA went into solution. And some of the rough bacteria were able to pick up the genes which confer the ability to make a capsule. And they were transformed. This is a real term in biology, especially molecular biology. If we say a cell has been transformed, it means it has acquired DNA, it has acquired genetic information in the form of DNA. Other workers went on to show that if you treated these solutions with DNAs, they no longer transferred genetic information. If you treated them with proteases, which killed proteins, they still did this, which was a demonstration that the hereditary material was, in fact, DNA. There would continue to be controversy. And people continued to say that DNA was too simple a molecule. They didn't know its structure, but they knew its components. And its components go back to our biomolecules section. And you'll remember that the DNA is a polymer, and it is made up of nucleotide monomers. And the nucleotide monomers, they consist of a sugar, a phosphate group, and then a nitrogen-rich ring structures, which we call the bases, of which there are 4 in DNA: adenine, guanine, cytosine, and thymine. So that much was known about DNA. But scientists looked at that and said, well, this is so simple. DNA can't possibly contain all of the information for all of the enzymes and everything that drive a cell. The information must lie somewhere much more complicated, like in proteins. And another set of experiments was performed by these workers, Hershey and Chase, Alfred Hershey and Martha Chase. And they were able to show definitively that DNA is the genetic material. They did it using a virus. I'm not going to go over the Hershey-Chase experiment. I want you to do it on your own. And you can submit it to me for 1% extra credit. I don't care, no longer than 2 pages. But as long as you clearly explain and demonstrate that you understand the Hershey-Chase experiment and why it was that it proved that DNA and not proteins conferred the genetic information. Okay, and you submit that to me, I don't know, after Thanksgiving. But please study this. because this is for you to study on your own, but it is examinable. Okay, so eventually it was accepted. And the DNA is the master genetic molecule. DNA is by no means the only nucleic acid present in the cell, as we'll hear, but DNA is the master code for everything that happens in the cell, because all of our cellular activities are driven by the action of enzymes. Most of our enzymes are proteins, and the DNA, in fact, contains a code for all of the proteins of the cell, including the enzymes. So this much is known. DNA is a polymer. It is the monomer that are joined together to make the polymer are nucleotides, and each consists, as I've told you, a nitrogenous base, a sugar, and then a phosphate group. I'll remind you about their disposition in a minute. Those are the four nitrogenous bases: adenine, thymine, guanine, and cytosine. Very, very early on, something extremely interesting, and at that time it was baffling, was noted about DNA. A scientist called Erwin Chargaff extracted DNA from many, many, many different species, lots of different animals, plants, all sorts of things, bacteria, and he looked at the composition of the DNA. And he noted that the DNA composition varies from species to species. So the proportions of these bases will vary from species to species. And this kind of diversity is underlain by a very important thing, which became known as Chargaff's rule. And that was that although the overall base composition varies between species, in any species you look at, the amount of adenine always equals the amount of thymine. The amount of guanine always equals the amount of cytosine. Doesn't matter what you're looking at. And this became known as Chargaff's rule. A always equal to T, C always equal to G. They had no idea why. They had no idea why that was the case at all. And it was only later on when we understood the nature of the DNA structure itself that it became clear why this was. So let's just remind ourselves here, this is like a base map, really, for the nucleic acids. Let's remind ourselves, first of all, here is the sugar here. I know that we're looking at DNA, one of the two DNA strands here, because that there is just an H. Remember that we number the carbons 1', 2', 3', 4', 5'. At the 1', there sits a nitrogenous base. At the 2' in DNA is a hydrogen. In the other nucleic acid, RNA, that we're going to talk about, there is an OH group there. This simple H here means this is a deoxyribose sugar. This is a ribose sugar, and it's deoxygenated, deoxyribose, because it's only got an H. The 3' site, remember I've told you, you have to go to bed at night saying 3' hydroxyl, 3' hydroxyl before you go to sleep, because especially today, you're going to learn out how important that 3' site is. It is the only reactive site. So that's the 3' site. Here is the 5' site here has a phosphate attached to it there. So notice something immediately. We make a polymer of this, and here are all our nucleotides all joined together. And oops, sorry. I just need to move something out of the way that you can't see, I believe. Okay, so let's just notice up at this end here, there's a phosphate group there. come down to the other end, and look, we have an exposed OH group. Where is it? It's at the 3' hydroxyl. That is the 3' hydroxyl. This is the only reactive site on this molecule. If I want to add anything, if I want to add another nucleotide, this is the only place I can do it. It can only come in here. It can't come down here. It can only come in here and react there. So this is one side of the DNA molecule, which is actually a double strand. But immediately notice we have a very strong backbone, and it's made of alternating sugar, phosphate, sugar, phosphate, sugar, phosphate. At this end here, there's the phosphate group, which is at the 5' site. We call this the 5' end of this strand. At this end here, we have this 3' hydroxyl. We call this the 3' end. So there is a backbone, and then poking out from the backbone like this, there are bases. And these bases can be present in any order. That's not to say that the order is random. It's not. It's very carefully structured, because locked into the sequence of bases here, as we're going to find out, is the code for the proteins that will eventually be made when all these processes are all said and done. Just because I've got an adenine here doesn't mean to say I have to have the cytosine there. Okay. If I got adenine here, I could have another adenine there. I could have another adenine there. I could have added adenine, thymine, guanine, whatever. What is present is determined by the code locked into the DNA. And the code ultimately is for amino acids, and it puts amino acids together in a sequence, which will produce a polypeptide that will go on to form a functional protein. So it was a long, long time before the actual structure of DNA was delineated. And they had lots of workers had lots of ideas about how DNA was actually structured. And they certainly had a very strong intimation that this was a polymer, and that the polymer somehow was made up of these nucleotides joined together, but they had no idea really how they were joined together. Was it one strand, two strands? Was the backbone inside? Was it outside? Whatever. They had no idea whatsoever. Yeah, DNA was very, very difficult to work on. It's not an easy molecule to work with. But there were certain workers who were exceptionally good at it. And this lab here, Maurice Wilkins and Rosalind Franklin in London, they actually were able to crystallize DNA, which is not easy to do. They were able to crystallize it and expose it to X-rays to get a picture drawn by what is known as X-ray crystallography. And I'll give you an analogy. X-ray crystallography has been compared to shining light up at a chandelier hanging from a ceiling, then mapping all the spots, the reflected spots, and then afterwards being able to recreate the shape of the chandelier just from looking at interpreting those spots of light reflected from the crystals of the chandelier. That's a fairly good analogy for shining an X-ray beam through a crystal and seeing what happens to the X-ray beam. It gets diffracted by the atoms and casts a pattern, which you can then try to interpret. So this was a scientist called Rosalind Franklin. She worked in this lab with Maurice Wilkins. She did not get on with him at all. She didn't, and unfortunately, that meant that her work wasn't quite as widely publicized as it ought to have been. But these other workers, Watson and Crick from Cambridge, they didn't really collaborate with this lab, but they visited periodically. And on one of those visits, they saw this picture which Rosalind Franklin had managed to produce from a DNA crystal. And almost instantaneously, they had a very good idea about how the DNA molecule was structured. And they became convinced that DNA was not a single strand, it was a double strand. And that, in fact, the backbone was on the outside, not on the inside, which many people had thought. Lots of people thought there was a backbone of nucleotides down the middle, and then the bases all stuck out from there. That seemed logical because the cell has got to be able to read those bases. It's got to be able to interact with the molecule. So it seemed logical that the bases should be pointing out, but they don't. They point in. And this was Watson and Crick's great intuition, especially when they saw this picture. And the picture also gave them the width that the two strands of DNA were apart from one another. And in another leap of intuition, Watson and Crick realized that distance was constant across the middle of the strand, of the DNA strand, and that there had to be the same number of rings across the strand always. Now, what I'll explain that to you like this. There are two kinds of base: purines, which is adenine and guanine, and they are two rings, like this. And then cytosine, thymine are pyrimidines, and they are a single ring. Watson and Crick looked at this double, they reworked it out, but they had a double strand of DNA. And the bases faced into the middle like this. And the bases had to match up somehow in the middle. If you put two purines across, you'd end up with 4 rings. 1, 2, 3, 4. It's too wide. If you put two pyrimidines together like this, it'd be too narrow. Okay. The only way to maintain a constant width all the way down was to have 3 rings, 1 purine and 1 pyrimidine. And that fitted with this X-ray crystal picture that they had seen. The next burst of intuition from Watson and Crick was that the 2 strands of DNA were anti-parallel. Remember, I told you that on the one end we had a phosphate, on the other end we had a hydroxyl group. They reasoned that one strand running that way would be matched by a strand running the opposite direction. So where we had 3' at the bottom of the strand we originally looked at, the matching strand would have 5', and then the opposite at the top. Not only that, they also reasoned because they said a purine must match to a pyrimidine. They suddenly thought back to Chargaff's rule, which said there was always the same amount of adenine as there was thymine. There was always the same amount of cytosine as there was guanine. They suddenly realized that this explained Chargaff's rule. If adenine always matched to thymine, purine to pyrimidine, and cytosine always matched to guanine, that would explain why, no matter what DNA you looked at, you would always have as much cytosine as you would have guanine, as much adenine as you would have thymine. It's because they had that rule says they have to bind like that, A to T, C to G, always, no matter the order on one strand. It always is matched, A to T, C to G on the other strand, and in an anti-parallel fashion. I'll show you a diagram in a minute, which will make it a bit clearer. Just let me check that I've got everything. Yes, I do. Okay, so have a look here, and you'll see exactly what I mean. Here's just a single strand, as we saw before, when it said, okay, this is like the base skeleton of nucleic acids. So here we have here's our deoxyribose sugars in the backbone, joined to one another by these phosphate, sugar, phosphate, sugar, phosphate, sugar groups. At this end here, we have the phosphate here. It's non-reactive, you can't react anything to it. Here's the phosphate group, that is the 5' end of that strand. This end here is the hydroxyl group, the 3' hydroxyl. This is the 3' end. Here are the matching strands. Look, these nucleotides have adenine, cytosine, guanine, and thymine as the bases facing into the center of the molecule. And they are matched. They are matched adenine to thymine on this nucleotide, cytosine, guanine on this nucleotide, guanine to cytosine on this nucleotide, thymine to adenine. So they are matched T to A, C to G, G to C, A to T. No matter the order on one strand, it must be matched by its appropriate pair on the other side. But look how they run. We've got 5' to 3' here, and we've got 5' to 3' there. This strand, if you want to think of it, is upside down in relation to that strand. That is what we mean by saying there it is anti-parallel. So the orientation is 5', 3', 5' to 3' on the other strand. That's the only way that you can put these bases in, because they have to bond. These little dots here are hydrogen bonds, 3 between guanine and cytosine, 2 between thymine and adenine. The only way you can get them there is to turn the one nucleotide inverted relative to the other nucleotide, which means that we have one strand running in one direction, the other strand running in the opposite direction. They are anti-parallel. And that is the beauty of this molecule. It is indeed very simple. It's also extremely strong. This backbone here is very strong. These are very strong covalent bonds. They're called phosphodiester bonds, and they are extremely strong, not easily broken. But these bonds here across the middle are hydrogen bonds. They are weak bonds, but DNA is a very long molecule, and it's a very big molecule. So there are many of these. And the DNA molecule is actually very stable. But it's important to note that these are easily severed, which is critical for the functioning of DNA. These hydrogen bonds are easily severed, but they are easily made again. And in fact, I'll just give you an example. We could take even a very large DNA molecule like this, and we can sever these bonds quite easily by heating the molecule. We say the molecule denatures, and these bonds split apart. These bonds stay together. And if we then cool a solution of DNA, it will come together very faithfully to its original matching, because it's driven by these requirements. So and it will come back together, and we can, we'll find out later, we actually use this in some scientific techniques. We split it apart and bring it together, split it apart, bring it together. These bonds are, so you learned now, the bonds can be severed by heat, certainly, but that doesn't happen in nature. That's our experimental way of doing it. But in nature, these bonds are easily separated by enzymes, as we'll find out now. And they often are. Okay, so let me just, I think you've seen this diagram before when we talked about nucleic acid structure. Nonetheless, let's have a look again. DNA, the molecule, what is incorrect, what is incorrect here is that we've flattened the molecule just for convenience sake and put it into 2 dimensions. But in actual fact, every sugar phosphate bond is at a slight angle in space. And this casts the backbones into these helices. And when you put the 2 sides of the molecule together, then you make a double helix. So the DNA is a double helix, and you can see the orientation. Here's the 5' here, end of this strand, follow it all the way down, and we end up with 3' at that end, 5', 3'. They are anti-parallel. The bases across the center here must match A to T, C to G, G to C, T to A, but the bases can be in any order. Any order of bases is possible. Again, it's not that they just go in randomly. It's just to make the point. Any sequence of bases is possible. This is what makes DNA so valuable as a store of information, because the number of combinations that you can have is infinite. Just with those 4 bases, having them go in, being able to go in any order means we have an infinite number of codes, of code words that we could use to describe what we need to. So there's a sugar phosphate backbone, the hydrogen bonds between the nitrogenous bases into the middle. DNA is a double molecule. We need to think, though, about the other important nucleic acid that we're going to hear a lot about when we talk about this, about molecular inheritance and everything else, and that is RNA. RNA differs. It has, instead of a deoxyribose sugar, it has a ribose sugar. At the 2' hydroxyl, at the 2' carbon site, it will have a hydroxyl group. So it will have, and not just a hydrogen, but OH group, and that is ribose. And RNA is fundamentally a single-stranded molecule. What is a little bit difficult to conceive of, though, is that it's a very flexible molecule. That backbone is very strong, but it is also very flexible. In DNA, it is cast into this shape here and is stabilized by the bonds across the center. But in RNA, the molecule can fold back on itself. The single molecule can fold back on itself, and it will match base to base, obeying much the same rule as DNA. Have a look here, and you'll see here's guanine, cytosine, guanine, cytosine. But RNA does not use thymine. It uses uracil. Uracil is very similar to thymine, but its place in RNA is actually a sort of a label for the cell. The cell can identify what it is dealing with because it has uracil, and the cell recognizes that as RNA, not DNA. But otherwise, uracil behaves exactly the way thymine does. It bonds to adenine. So it's uracil is also a pyrimidine. And we got purine pyrimidine, etc., just as we did with DNA. The only thing is that these bases here are actually on the same RNA molecule. Have a look here, like a roller coaster. This is one molecule. It's a single molecule. In fact, its 3' end is there, its 5' end is there. This is an RNA molecule, which we're going to hear a lot about. This is tRNA, or transfer RNA. It's like an upside down L shape like this. And it's all of these base pairings here are between different areas of the same molecule, not another molecule like in DNA. When that happens, when these base pairings happen, they cast this molecule into a particular shape in space. So it has a particular 3-dimensional shape, which is conferred on it by all of these bondings. That is typical for RNA. Very often, RNA can perform a structural function. We're going to find out, for example, in the ribosomes. The ribosomes are actually made of RNA, big RNA molecules, which are convoluted into 3-dimensional shape because of their base pairing across the same molecule, pulling the molecule into that 3-dimensional shape. So this is transfer RNA. All right, so the one thing that should be immediately apparent, but I want to really emphasize it, is let's just go back to, let me just go back here and say, if I was to destroy one of these DNA strands, I would immediately be able to recreate the matching strand on the other side because of this rule here. Let me just take out, let's just take these, pretend these bases have been obliterated on this strand here. They've been lost for some reason or another. We can recreate it. We know that that must be C because this is G. We know this must be G because that's C. We know this must be T because that's A. We know this must be G because that is C. Knowing one side, you can always recreate the other. What you need to be careful of when you see this kind of exercise is to take careful note of the direction that the strand is running. Okay, it can be very important, as you'll hear in a minute. Okay, so if we know one strand, the other one can be constructed. What is this? What is it here? Take a minute. Three prime T A A T C G G. Okay. It has because we have to match G to C, G to C, G to C, T to A, etc. Same here. This one, 5 prime, C, C, G, T, C, G, C, A, T, 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 T. Okay, knowing one strand, you can always recreate the other. And this is really important because it means that if we split those strands apart like this, we can recreate each strand. We'll have to do it in opposite directions because they run anti-parallel. But if I've got a DNA strand and I separate the two halves, I can recreate the other half immediately because I know what the sequence is on one side. It determines what the sequence is on the other side. Okay, just to one thing to say, if you have a look at the purine pyrimidine matchings, then you find adenine and thymine. Here there are 2 bonds here. There are 2 hydrogen bonds. Guanine and cytosine, there are 3. And this is actually a stronger bond than this bond here. That only becomes important when you are interested in molecular biology. This has some important implications, but this is a stronger bonding than this one. This, to emphasize that this one needs a little bit more heat to split apart than this one. This one is more easily separated. But again, 2, 1, 2, 1. And it's a beautiful molecule. It is cast into this spiral in space. There's a full turn every 10 base pairs. That's 10 base pairs to 1 for 1 turn. And you'll notice something, and that is that there's this groove here is wide, and this groove here is narrow. So you've got a wide groove there, and then a narrow groove here. And surprisingly enough, it's a lot of the cell examination of DNA is here in the this narrow groove there. Okay, so the first thing we need to think about is that we discussed the processes of mitosis and meiosis at great length. And one of the critical steps that takes place in cell division is that before any of the stages of cell division arise, the cell replicates its entire genome. We started off looking in a eukaryote. We saw that we started off with 1 chromatid, and that chromatid was entirely replicated to provide us with 2 chromatids for much of the cell's life. It has 2 identical chromatids on each chromosome. So this is a massive undertaking. In a human cell, we have 46 chromosomes, for example, all of which must be faithfully replicated. And the other thing is that when a cell is busy replicating, a lot of the cell function actually stops. So we have to replicate really quickly, but we need to do it really accurately. It has to be done so that as few mistakes are made as possible, because a mistake in replication could introduce a mutation, which can have effects on the functioning of the cell. So replication is tremendously important, not only in mitosis, where, for example, we are replacing one cell with another or contributing to growth or whatever, but in meiosis as well, because in meiosis we are creating the gametes, and that is how we are going to hand on information to progeny. So the information handed to the progeny has to be as accurate as possible. And in order to do this, we need to actually double up the DNA, and that is a process known as DNA replication. And one of the beauties of the Watson and Crick model was it explained very simply how the basis of DNA replication worked. And the fundamental thing, of course, is that since they match base C to G, T to A across the 2 strands, each strand of DNA acts as a template for the other strand. If I split them apart, I can recreate the strand on either side. And that immediately gives you a model. It's good. If I can split them apart, recreate the strands, and I will have made 2 copies, exact replicas of the parental molecule. So this is the very basis of it. Okay, here we have the parental molecule, and we're going to separate them like this, and then we're going to build the new ones. Okay, here they match A to T, C to G, T to A to T, G to C, split them apart, and we can now recreate A to T, G to C, A to T, T to A, etc. So there's a couple of things to notice here, and that is, yes, we bring these bases in, but look, they come in and they automatically are anti-parallel. they automatically replicate the parental molecule. There it is there, exactly the same. And here it is here, exactly the same. So we now have ended up with 2 replicas of the original. But what's also important to notice is that what we end up with is one old strand here. There's the other old strand there, and a new strand. So when we replicate DNA, we end up with the replicated strands consist of one parental strand and then a new strand, parental strand, new strand. And this is termed semi-conservative. It's a semi-conservative replication because we end up with half of the molecule conserved from the parental and half of it is new. These we call the daughter strands. These are that for this purpose is the parental strand. All right, so if you want to follow it through, there is an experiment described in your book called the Messelson-Stahl experiment, which you can read through. I'm not going to go through it in detail, but there was some argument about the nature of the replication of the DNA molecule. One model suggested that it was entirely conservative. So when the DNA replicated, we'd end up with just the parental strand at the end, the parental strand as it was, and then an entirely new strand. The other model was the semi-conservative, which is what we actually have. Each new strand, half of it is parental and half of it is new. Then there were some people that argued for what was called a dispersive model, where it was kind of a mixture. You couldn't, the strands were all mixed up, but they replicated one another. The Watson and Crick, their model said that the semi-conservative was right. And then these workers, Messelson and Stahl, they actually positively demonstrated that the semi-conservative model was the correct model. You can read through that if you wish to do so. All right, so now we've got to think about this huge task ahead of us, at least a huge task for ourselves, of replicating the entire genome inside the nucleus. This is a huge task because there is so much DNA. We have meters. If we were to string out our DNA and one long line, we would have meters of DNA in a single cell. It is very carefully packaged inside the cell so that it fits in and that it is easily used. But you must realize that during replication, a lot of the function of the DNA stops because you can't use the genes. You can't read the DNA during the process of cell division. So it has to be done really quickly. And there's so much of it that it has to be, keeping the accuracy requires the interaction of numerous different enzyme systems. But this is a critical time in the cell's life, the processes of DNA replication, because it has to be done accurately. You cannot continually accumulate mistakes, mutations every time you reproduce a cell. So we'll hear about some of the ways in which the cell accomplishes that now. There are more than a dozen enzymes and some associated proteins which are involved in DNA replication. Most of the processes of DNA replication were elucidated in prokaryotes, where it was rather simpler to work it out because they have a much smaller genome and they only have one chromosome. But in actual fact, the same processes apply with a little bit of modification to all organisms. In most organisms, follow exactly the same kinds of rules and everything that are present in prokaryotes. So replication always begins somewhere, and that somewhere is actually a specific place. It's not just a random thing. It is called an origin of replication. And in the prokaryotes, this was another advantage of looking at the prokaryotes, they only have one origin of replication. And it's attached to the cell membrane. And replication begins there, and it spreads out from there around, in prokaryotes, around the circular chromosome. But in eukaryotes, we've got so much DNA that if we were to just have a single origin of replication on each chromosome even, replication would take far too long. So in eukaryotes, this is one difference to the prokaryotes. In eukaryotes, there are many, many origins of replication on each chromosome, so that replication can be done really quickly. When you have an origin of replication, then replication begins on either side of that origin and spreads out from there in a particular shape that you'll see in a minute. So here are how the origins of replication work. This is a beautiful electron micrograph of a replicating replicating bacterial chromosome. And what you notice is there's a Y shape, and that is called the replication fork, and this is how it's produced. Here in bacteria, remembering that DNA is a double strand, as shown here, there is a single origin of replication in bacteria, and it's going to spread out from there. So replication is going to proceed in one direction here and in the other direction on the other side. And then eventually the two meet in the middle and you end up with two separate new chromosomes. This is what we mean by the replication fork there. There. That's the replication fork. There it is there, and there it is there. So on the bacterial chromosome, that's quite easy to understand. On the eukaryote chromosome, there are several origins of replication. So here's an origin here, and here's the replication fork on that side. Here's the replication fork on that side. Replication is proceeding in that direction and in that direction. And it's eventually going to meet up with the replication fork coming in the opposite direction there. And here they are here. You can see replication forks there and there, there and there. This is on eukaryote chromosome. Remember that a chromosome is one single strand of DNA, extremely long in the eukaryotes, carefully packaged into the chromosome. So, okay, I'm just going to flip to the diagram to explain. So we are going to just think now about how this procedure goes, remembering that we've started back here somewhere at an origin of replication. We have split the two strands apart. That separation of the strands is governed by an enzyme, and the enzyme is called helicase. Here it is here. Helicase runs down the strand, and it severs the hydrogen bonding between the bases. So now we have single-stranded DNA here and single-stranded DNA here. And this is the direction that the helicase is going to run. So the opening up is going to take place in this direction. DNA is this complex double molecule, a double helix in space. Rather like a string. If you separate strands of string and you pull it apart, downstream of where you're opening up like that, you introduce tremendous stress on the rest of the helix. This is very dangerous for DNA. It can cause damage to the DNA to be continually tightened like this. And there is a set of enzymes. Here they say topoisomerase. In fact, there are a couple of, you can just remember topoisomerase, and I'll just tell you that there are a couple of enzymes that perform the same function, all of them topoisomerases. In any case, the stress downstream here is relieved by this enzyme, topoisomerase. It actually can cut DNA carefully, allow it to unwind, and then it gets fixed again. So we've got helicase opening up, topoisomerase relieving the stress and tension. There is one problem, and that is in many cells, especially in prokaryotes, do not like to see single-stranded DNA. As soon as they see single-stranded DNA, it says to them, virus, and they will attack it. So what happens is that as helicase splits these strands apart, they get stabilized, and they're stabilized by a set of proteins called the single-strand binding proteins. And the single-strand binding proteins, they protect it from digestion, but they also stop it from reassociating, because it'd be very easy for these bases just to reassociate with one another. Instead, they're kept apart by the single-strand binding protein. Fine. So now here comes a really important rule. And that is, unfortunately, we can't replicate DNA just straight away. DNA is replicated by an enzyme called DNA. It's a DNA synthase. I mean, it's going to make DNA. So it's actually called DNA polymerase. There are a couple of DNA polymerases, but this one that we're going to be talking about on this strand here is DNA polymerase 3. DNA polymerase 3 is a wonderful molecule. It has all sorts of problems, but it can't just fix onto the single strand and start reproducing, making another DNA strand. Instead, it has to have a short length of RNA laid down. We start off with a little length here. It's not just 2 bases, by the way, it's a bit longer than that. It's laid down by an RNA polymerase. It has a special name here. It's called the primase because it's laying down a primer. And it's got the primase comes along and it lays down a short length of RNA so that we have a hybrid molecule, DNA and RNA, for a little while. Once that primer is laid down, then DNA polymerase 3 can latch onto this, and it will carry on happily making DNA after that. Oh yes, let me just point out something to you. This is also like an important thing to note. Remember what I said to you. 3' hydroxyl. Why emphasize that the whole time? Because that's the only place we can add nucleotides. We can only add nucleotides at the 3' hydroxyl site. Let's just have a look at this little bit of RNA here. It's got a 5' end and a 3' end, just the same as DNA would have. All the nucleic acids have that rule, that feature. Remember, the 5' end runs this strand runs anti-parallel to this strand. So look here, we have the 3' end of the DNA that we're reproducing. So we have the 5' end of the RNA strand here. This is the 5' end of our primer. The only place we can add nucleotides is here at the 3' end. So we are going to grow this strand from 5' to the 3' direction. We're going in that direction. Why? Because we always have to add here at the 3'. We can't add here at the 5'. We have to add here at the 3'. It's going to grow in this direction. In this case, it's growing in the same direction as we're opening up. So once we've laid down a little primer there, and the DNA polymerase comes in and starts to make the strand, it can go for thousands of bases. It can go for ages and ages and ages, following behind the helicase as the helicase opens up. So the DNA polymerase will lay down the new DNA strand. This goes very fast, and it's about 500 nucleotides per second, a little bit slower in eukaryotes, 500 nucleotides per second in bacteria. It's extremely fast, and purposefully so, because the cell needs to get this over and done with as quickly as possible. So this is an energetic process. It requires an input of energy, but fortunately, there is an inbuilt source of energy, and that is that the free nucleotides inside the cell are called, here it is here, it's actually called a nucleoside triphosphate. Just remember that triphosphate bit. That's what's really important. Here's the free, here, the free nucleotide here. There's the sugar, here's its base, this is a thymine. And it's free inside the cell. It has 3 phosphate groups. Do you remember what ATP looked like? ATP was an adenine, right, with 3 phosphates on it. That's our main source of useful biological energy is ATP. And the energy was lodged in these bonds. It's exactly the same for any of these free nucleotides. They come in to join in this form, in a triphosphate form. And that phosphate is severed when they make this bond here. They're going to make a bond between this hydroxyl group here and this phosphate here. That to that, this is going to be freed, and the energy is going to be harvested and used to make that covalent bond. So that each of them is carrying their own new energy source with them. And that's utilized by the DNA polymerase to make this phosphodiester bond here, the new phosphodiester bond. And here, notice we have another free hydroxyl group ready for the next base to come in, for the next nucleotide rather to come in. The base is matching here. So the next one is going to be carrying guanine, and it will come in as a guanine triphosphate. 2 phosphates will be severed, and the energy will be harvested to form a new phosphodiester bond here. Okay, so when I explained that we put on this strand here, we put on our RNA primer, then the DNA polymerase 3 will come in, and it will begin to match in bases and will run in the same direction as the strand is opening. So it just carries on and can carry on for tens of thousands of bases before we need to start again, or before it runs into the next replication fork, which is in eukaryotes, will be coming in the opposite direction. This strand here, because we're laying down new strand in the same direction as we are opening, we call this the leading strand. And on the leading strand, you just have to lay down 1 primer, and then you can carry on for a very, very, very long time before you need to start again. Remember, this is DNA polymerase 3. What happens on the lagging strand is a little bit different. The lagging strand is the strand opposite, where the DNA has to be made in the opposite direction to the way that the strand is opening. Now, this is this I need to explain carefully, because it's always a little bit difficult to conceive of. Here's the problem. In order to lay down the primer, I actually have to have bases available. Okay, so let's just say, come back here. Let's say now I've opened this up here, okay, and I'm started my leading strand there. I've opened this up here. Well, I have to have enough strand here open to be able to put down the primer. So I'll put down my RNA primer. But look at the direction. If this strand is running 5' 3' there, then we're going to be running, our primer is going to be running 5' to 3' in this direction, in the opposite direction. I'm going to have to start my building my DNA strand down here somewhere, lay down primer here. Here's the 3' would be the 3' end of the primer. And I'm going to start laying down my DNA there. But in the meantime, I've opened up, I will have opened up behind here for this to be laid down, for the leading strand. So now I've got a whole area which is open. What am I going to do? I'm going to lay down another RNA primer. And then I'm going to make DNA until I bump into into the RNA primer that was laid down before. Okay, have a look. Let's just walk this through, and you'll see here we go. Here we lay down our primer here, and now I'm going to be beginning to lay down DNA in this direction, right? This is on the lagging strand. The opening is going in that direction. But I'm laying down in this direction. So I lay down a primer, make my DNA, and boom, what happens? I bump into the primer that I laid down before. Meanwhile, a whole area has opened up behind. Here's my strand here. Now I've opened up a whole area behind here. I've got to lay down another primer. Here I lay it down here. And I run, now the DNA comes in, DNA polymerase comes in, and it makes the DNA matching strand. Boom, it runs up against my old RNA primer. So what has happened on the leading strand here, in this direction here, the new DNA is laid down continuously as a continuous strand. But on the lagging strand here, the new DNA is laid down in fragments. And the fragments consist of RNA, DNA, RNA, DNA, RNA, DNA. And they are not joined to one another. They sit as separate fragments. So what happens on the lagging strand is that we have these fragments which are called Okazaki fragments. And they arise simply because we are laying down in the opposite direction to the way that the strand is opening. So we have to continuously relay down. We continuously have to put down new RNA primers that are then served as a basis for DNA polymerase III to make the new strands. So in the end, what happens is we'll end up with a whole lot of these hybrids, RNA, DNA, RNA, DNA. And somehow or other, this all has to be stitched together. The way it's stitched together is that another DNA polymerase comes in. This is DNA polymerase I. And DNA polymerase I is able to digest the RNA primer and lay down DNA bases in its place. So that is exactly what it does. It digests out the RNA primer and it lays down DNA base in its place. The only problem is that when it finishes, can you see here? When it finishes, it leaves a gap there. It can't make that last bond there. And another enzyme called DNA ligase comes in and heals up all of these. This is called a nick. The DNA ligase comes in and heals up all of the nicks so that we end up with one single continuous strand. Okay, in summary, the leading strand is laid down in the same direction as the DNA strand is opened, as the parental DNA strand is opened. It only needs one primer. On the lagging strand, we have to have enough open to lay down a primer and then do the replication. So we end up putting down repeated primers. As the strand opens up, we put in another primer, recreate, etc., etc. We end up with a set of Okazaki fragments, which are a mixture of RNA, DNA, RNA, DNA. That RNA is digested out by DNA polymerase 1, and all of the little gaps left behind are healed up by DNA ligase. The DNA, by the way, this primer here is also taken out by DNA polymerase 1, and ligase heals up the little nick that is left behind. So that is fine. Here's a replication fork. On one side of the replication fork, leading, on the other side, lagging. But have a look at the other replication fork. This replication fork, the direction of opening is in the opposite direction. It's in that direction there. Okay, so now this strand here is the leading strand, and that strand is the lagging strand there. But it doesn't matter. The enzymes still complete each of these strands, and they are still healed up by DNA ligase when the whole process is completed. Okay, so make sure that you understand this really, really well. There are some little animations and things here for you that will explain it even better. Here's the whole idea put together in one. Leading strand here, old parental strand is the template. Here's the other parental strand here. Leading strand, one continuous strand laid down by DNA polymerase 3. This strand here, repeated RNA primers, repeated replication by DNA polymerase 3. Then DNA polymerase 1 comes in, eats out the RNA primers and replaces those bases with the DNA. And finally, DNA ligase links the whole lot together into a continuous strand. And what do we end up with? Where we had one parental strand, we now have half of it is parental, half new, half parental, half new, and we have replicated the parental strand. Okay, here's something to think about, that a prokaryote like E. coli, if you put it into rich medium and you give it a good temperature like 37 degrees Celsius, it will replicate its DNA every 20 minutes. And the rest, it'll do that, replicate its DNA really, really quickly. And the rest of the time it devotes to growing the cell so that it can divide off and form 2 new cells. Every 20 minutes, it will replicate like this, and it will do it accurately. This is a question to ask. It is possible during the process of replication to actually put in the wrong base. And this is potentially this potential problem. One of the ways in which this does happen to the bases are put in incorrectly with a certain regularity. You can work out how often it happens. But one of the amazing things about DNA polymerase 3 is that it is able to proofread. It is able to check the bases. It will check the bases against the parental bases. As it lays down bases, it will check that it's put in the correct base. If it has not put in the correct base, it will back up. It will digest out some bases, and then it will repeat itself and move forward again, laying down the correct bases. And in this way, we actually reduce the error rate by 1,000-fold. and most of the error is entirely avoided. If other errors do occur in this, then there are other repair mechanisms that can come in, check the DNA, and attempt to repair it, and make sure that the correct bases are put in. Okay, here's a summary for you of all of the different enzymes that are involved in replication. Okay, just one consideration, especially for eukaryotes, that is really important. We have an enormous amount of DNA present inside a nucleus, and that the DNA can't just lie there in a random sort of fashion. It has to be carefully packaged, and it has to be packaged in such a way that the genes can be accessed really easily when they need to be. Some genes are read off continuously. We call them household genes. They are for stuff that we need the whole time. But many genes are only needed periodically, and the cell has to be able to access them, and to access them easily, they have to be carefully packaged. The DNA has to be carefully packaged in a predictable fashion. So eukaryote DNA is linear. The prokaryote DNA, the chromosome is circular, but all of the eukaryote chromosomes are actually single DNA molecules of great length. They are associated with a set of proteins called the histone proteins. That they are actually, the DNA is actually wound around individual histone proteins, rather like beads on a string. And the cell can then take those histone proteins with a little bit of DNA wound around them. The cell can then manipulate those histone proteins in various ways to package them in a predictable fashion. For most of the cell's life, as you know, we look inside the nucleus, you cannot see the chromosomes. Instead, the DNA with its associated histones is unwound. And when you look at it in the microscope, it just forms a granular background, which we refer to as chromatin. And chromatin is a complex of DNA with its associated histone proteins. And then those histones can then be wound themselves in various ways at different stages of the cell's life. During interphase, they are relatively unwound. And the association, the little histone with its DNA around it is called a nucleosome. And the DNA winds twice around each nucleosome. And those histones, those nucleosomes, here they are, here, here. These can then, for much of the cell's life, they're like this. Okay, they're kind of relatively unwound. But as cell division approaches, so the cell begins to wind these histones together and then begins to spin them around and around and around into these complex shapes, which contract the DNA denser and denser and denser until they're actually visible as the chromosomes here. You don't need to remember the details here. All I want you to remember is that the DNA is wound around histones, and the histones themselves can then be manipulated and packaged depending on the stage of the cell cycle that there is. During interphase, when the nucleosomes are relatively unwound, they are still present in the cell in a predictable fashion. They are actually carefully placed inside the nucleus. So that they don't interfere with one another. You can actually go to the nucleus and you can actually stain for particular chromosomes. And this is this marvelous this marvelous micrograph. And you can actually see individual chromosomes. They're each stained differently. And here they are unwound to occupy specific areas of the nucleus. They do not encroach on one another's territory because they contain separate genes that need to be accessed. They are kept quite separate from one another. As cell division approaches, all of this unwound chromatin is spun down into the chromosomes. And here you can see the individual chromosomes themselves. You can actually see the different colors of the different chromosomes, but this is them in their interphase form here. Okay, so that is it for cell replication. This will be included in your test on Monday, so please make sure that you study carefully. And don't forget also that on Friday, Professor Bachu is doing a review session on Friday afternoon from 2 to 5. If you are not able to attend that, in actual fact, she will be recording it as well. So it will be available to you over the weekend. Okay, and with that, I'd say thank you.